A very pleasant morning to our esteemed management, our Secretary Sri Lalit Kumar Oj, our Principal Ma'am, Dr. N. Sujata. It is a pleasure to welcome you all to the grand finale of our international seminar on emerging technologies in chemistry. It is fitting that we conclude with a session that looks directly into the future of who represents a global reach of Indian scientific talent. Dr. Balasubramaniyan, Shakti Institute for Chemical Reaction Design and Discovery, WPI-ICReDD at Hokkaido University develops AI and machine learning models to predict chemical reactions and integrate them into robotic synthesis pipelines. His academic journey begins right here in Tamil Nadu with a BSc and MSc chemistry from Bharatdasan University and an MPhil from Gandhigram Rural Institute. He later earned his PhD in pharmaceutical technology from Anna University, where he focused on designing novel analogs to fight drug-resistant bacteria. Before moving on to Japan, Dr. Balasubramanian held significant roles in global industrial research, including serving as a senior research scientist in PhD with 17 high-impact publications in journals like Digital Discovery and the Journal of Molecular Structure, covering everything from SARS-CoV-2 inhibitors to environmental carbon chemistry. What makes Dr. Balasubramanian's presence so special today is CREPE for virtual product enumeration and pioneered AI-driven color atom drug design strategies. If I were to read out Dr. Balasubramanian's entire list of achievements, certifications, and I even started a map of global excellence. For our students, he is a living example how a foundation in chemistry can evolve into a career that spans programming in Python, molecular docking, and advanced MD simulations. So we are honored to have you join us from Japan.
So, Dr. Balasubramanian, sir.
Hello, sir.
Sir, you can take over the session, sir. There is some technical problem.
Yes, sir. Actually, I could not hear the voice. Maybe the.
Yeah, they have been disconnected from the line. You can take over the session, sir.
So my voice is clear. and my presentation is
Sir,
okay. My voice is audible from, I think, my,
little louder, sir, please.
hello.
Yeah, little louder, sir.
Yeah, my voice is audible.
Yeah, audible, sir. You can proceed, sir.
Okay, thank you.
Thank you.
Okay. So, good morning to all. I'm BALASUBRAMANIYAN. I'm doing a postdoc research in the Institute of Chemical Reaction Design Discovery, the Hokkaido University of Japan. So today I'm going to present the computer-aided drug design and drug discovery using a free web page, free web sources like that. Which means currently the most of the researchers in chemistry field or pharma companies, they are using the computer-aided drug design to fast discover the new drug. But most of the computer-aided drug design approaches based on the highly costly softwares. So it's very difficult to learn everyone to understand what is the computational chemistry and how we use the software like that. So today I'm going to explain what is computer-aided drug design and how we can do by free webplanes tool like that. So most of the countries and research labs, they discover the new software. They publish free online sources. So everyone, they go and learn and do your own research and get the results like that. So that's what today I'm going to present this. What are the link available and how we can design the potential research on these computer-aided drug designs. Okay. So first I will explain very basic examples. Um, we know that how our medicine is working in our human bodies. So whenever we are taking the medicines, for example, antibacterial drug, it will directly go to the human intestinal organs and it dissolve and mixed with the blood, and it going to the entire body. Once it's find the suitable target, like the target means is the proteins. For example, if the organism, for example, I'm telling about the E. coli, it's a main reason for fevers. When I taking the ciprofloxacin or fluoxetine drug, so the drug going to the intestinal body and mix the blood, then it find the suitable target, which means each each of the organism have the specific proteins, which means that proteins involved for their functions. So these drugs, like these drugs, they have the specific target proteins. In the protein, the drug will going to bind the protein and stop the functions. This is the actual mechanism of antibiotics. So first I will explain what is the proteins. Protein is not a protein is made up of amino acids. So it's a valine, tryptophan, alanine. These are the amino acids. They linked chain. It's called peptide. The many peptide mixed with is called the proteins. So protein is a functional organism for the microorganisms. Like in our human bodies, liver, heart, like this, in each organisms, proteins are very important for their functions. So the drugs going to bind this protein and stop the function. This is the actual mechanism of drug mechanism. Okay. So I'm going to explain what is drug designs. Once you find the suitable target, what are the proteins involved in the disease? Based on the protein information, chemically we have designed the molecules. That is called drug designs. Okay, how the molecule going to bind the target proteins? See, this is one of the examples. So this is the molecules. The molecule maybe we generally call as inhibitors. The inhibitors, actually each protein have a specific gravity that hold. All the protein have a specific specific binding sites. So the drug going to bind the gravity site and block the activity. So like this, the drug going to bind inside the proteins. That is the drug docking process. This is like the inhibitor going to bind in the proteins. Okay, I'm going to show traditional drug process. Generally, it will take 10 to 15 years for a new drug coming to our hand. Initially, researchers select the disease and synthesize the molecule, and they check with the animal model and go for formulation and that drug approval. Then you go for clinical studies like phase 1, phase 2, phase 3. Then finally, they approve the drug approval. Then finally, it's called the medicine drug. So it will take 10 to 15 years for entire research to discover a new drug. But the recent years, that's what we are using the computer drug design to fast the process. In couple of years, like 3 to 2 years, we can potentially discover the new drug. That's what currently most of the research labs, they are focused on computer drug designs. So I'm going to show the one small video. It's okay. The video is not working. Okay. So okay, that's the video is not working. So this is a close view how this molecule going to bind in the gravity of the darker proteins. This is the image. Okay. How the drug molecule forming interaction with the target proteins. As a chemistry, we know that we studied what is van der Waals interaction, hydrogen bond interaction, pi anion interaction, pi alkali interaction. These are the main importance for drug binding with the amino acids. So I already told that protein is made up of amino acids. Like this, in gravity, many amino acids there. In this amino acids, the functional group of the drug molecules forming the hydrogen bond interaction, van der Waals interaction, pi anion interaction, pi alkali interaction. Like this interaction formed to form the strong bond with the amino acids. Like this, drug will be active in the proteins. Okay, currently what are the modern drug they are using? Currently they are using two type of methods. One is structure based drug design, one is ligand based drug design. Then identify the binding site, then go for virtual screening. Virtual screening means computationally they select the best molecule, then select the best molecule, then it go for new drugs. This is a shortcut way to discover the new drug. Okay, this is all I explained. Okay. Okay. 1 minute. Okay. So why we are using computational aided drug designs? We can design a new molecule based on the computational techniques, speed up the reactions, and efficiently we can design the molecules. And we remove the falling drug before synthesis process. That's what everyone they are using computer-aided drug designs. Okay, what are the method currently they are using for drug design? One is ligand-based drug design, one is structure-based drug designs. So currently, most of the research institutes, they are mainly focused on the ligand-based drug designs. The reason is ligand-based means easily we can find the druggable molecules. Based on the druggable molecule, slightly we modify the functional group, that is called ligand-based drug designs. So for this is an example of ciprofloxacin. This is an antibacterial drug. From the antibacterial drug, slightly they are changing some functional group and they discover a new molecule. This is ligand-based drug designs. So most of the research lab, they are focusing ligand-based drug design, because from the ligand-based drug design we can easily design the active molecule without fail. But another method is structure-based drug design. Structure-based means if you find the new disease, easily you can select what are the proteins involved in the disease. So we can select the proteins. In the protein, we can easily find the binding site of the proteins. Based on the binding site informations, we can design the molecules. That is structure-based drug designs. These examples how we can design the molecules. Once you find the target protein and the gravity sites. Based on the gravity site information, we can construct the small fragments. Finally, we can make it a final molecules. That is a structure-based drug designs. Okay. Before going for structure-based drug design, we have to identify the what are the binding site in this protein, because each protein has several gravity sites. Many hole is there. Before the drug designing, we have to find the which one is the binding site of the proteins. Then only we can design the molecules. Okay. Next, I'm going to the docking. Docking is one type of in silico process. Once we design the new molecules, first we go for docking studies. Docking studies means it just design the molecules by docking with the target protein and check the docking score. If the score is very low, that molecules are higher binding activity. So later I will explain where we can do the docking and in silico studies by free of cost. Okay, this is advantages means time, cost, accuracy, and screening the best molecule before synthesis. These are the example drug. They are discovered the new based on this computer drug designs. Okay, these are the techniques currently we are using for computer drug designs. Like, for example, if you're focusing structure-based drug designs, docking, pharmacodynamics, de novo drug designs, and ligand-based means QSR, similarity search, and scaffold mapping, and ADMET process. These are the methods currently we are using to discover the new molecule based on the computer drug designs. Okay, these are the explanation. Now I'm going to explain what are the free computational tools available. Everyone they can do the own computational studies in using mobile or laptop without any software. Okay, before that before that we have to find the what are the databases there. Database is very important. From the database only we can extract them, we can download the molecules. These are the database like zinc database, ChemBL, and protein data bank. These are the database. From the database only we can get the information for computer drug designs. Okay. Sorry. Okay. This is one database. Name is protein data bank. In the protein data bank, all the disease target proteins are loaded in their proteins. So first, once you plan to do the research in the computational chemistry, you have to go first in the protein data bank. In the protein data bank, you can type what are the disease you want to do the research. You can simply type the disease, then it will show the what are the proteins involved in the disease. And from these proteins, first you can download this protein from this website, 3D proteins. Okay. So some people they want to identify the best molecule from already reported molecules from the database, because drug design, some people they don't have knowledge on the computer drug designs. So what they will do? They go for ChemBL database. From the database, large amount of non-reported activity molecules there. So from simply you can download the molecules for further process. This is one of the free website, free database. From that you can download the many chemicals based on this IC50 value. Okay, so sir, first I have downloaded the protein. I can download the large amount of molecules. How we can find the binding site of proteins? I don't have any software, so you can go this website, Prank Web. It's a free web server. Here you can load your proteins. It will show which one is the potential binding site where the drug going to bind the protein sites. So from the information, you can select your binding site of the proteins. It's a free sources. Okay, sir, I find the binding site. Then where I can go to the docking? I don't have software for docking. No problem. You can go for CB dock. It's one of the it's merged with the Autodock Vienna. It's give the highly accurate results. So no need any software. Just to go for CP dock and click the dock link. Okay, dock the link. This is the website front page. Simply load your protein and load your ligand, and it will select your gravity sites and submit. Within 5 minutes, you can get the docking score with the high accuracy results. So you can download the results from the docking. No need any software. Okay, once you download the software, download the docking results, then you can go how to download this software. This name is BioWare Disco Studio software. It's a free software. It's a visualizer. Once you download the software and install your laptop, you can load the previous results, this result, and see the binding interaction and 3D and 2D interactions. Easily you can enumerate the image from the docking results without any software. Okay. Okay. Sir, I could not install this software. No problem. You can go this app Boltzmann Lab. It's one of the protein view software. Once you download the docking results and load your docking results here you can see the protein actual structure and in 3D interaction, 2D interaction, and hydrothermal interaction, and the distance, everything you can get from these results. It's a free sources. Okay, sir, I done the docking. How I can do the further studies like Pharmac4, QSR, de novo drug designs, IDMT like that. So these are the free tool. And that, okay. This is another tool is Swiss target predictions. Here, for example, if you design the new molecule or you find the new molecule, you don't know what is the target of the proteins like that. It's very easy. You can go this website, Swiss target protein, and load your molecule and run this. It will show maximum percentage of where your molecule going to the bind in the proteins. From the name, you can easily download the protein from the protein data banks. Easily you can find the correct target of the proteins. Okay, sir, I want to design the new molecule like AI-based drug design. How we can do? I don't have any software. No problem. You can go this website, ligand-based drug design. Heisenberg developed AI tool. Here you can give some information like what type of drug you want design and how it should be like that. You can give the information. It will generate the new drug based on this your structure-active relationship like that. Okay, sir, I want to design the new drug based on this structure-based drug design like de novo drug design. Where I can do go this website. It's a eLead3. This click this de novo drug design website. This is a link. This is a page. Here, choose your protein and choose your starting material. It will generate the molecules inside the target proteins, and many molecules will develop. You can use this molecule for further process. It's a good online link. We can develop the potential molecule by structure-based drug design without downloading any software. Okay. Sir, I done this design, docking, everything. I want to do the QSR. Where I can do it means simple. You can go this website. It's 3dqsr.com. They have a free access. Once you log in this website, they will give you the free access. Here you can do the 3D QSR. It's the longest process. So before you are entering the QSR, you have to do some of the work for your list of the molecules. They have some ideas how to make it your molecules. once you follow this method, you can go this website and follow this method. You can easily develop the 3D QSR and predict the activity of your molecules. It is this one type of machine learning methods can get the good results. Even mobile also it will work. Okay, sir, I done this everything. I want to do the pharmaco research, like similarity search, the similar molecule I found, where I can do this. It's one of the free website. It's a Farmit. Here, once you load your protein and load your molecule, it develops the pharmacophore of your molecule, and also you to find what are the similar molecules related to your molecules. It will give this list. So from the list, you can easily find this what are the similar molecule reported or non-reported everything, and also you can download these three molecules for if you want to do the pharmaco studies. You can use this online tool and get this similar molecule based on your molecule data. This is one link how we can find the similar molecule search. So initially I load this one my molecule, and the online tool give the one molecule more close to my molecule like that. It is a good one for structure activity relationship and to find the similar molecules. Okay, another one is ADME tax. ADME means it's a toxicity prediction like adsorption, distribution, metabolism studies. This also you can freely we can evaluate. This is on website as ADME. Once you load your molecule, design the molecule, it freely predict the activity of the all the properties like lipophilicity, physiochemical properties, drug-like properties, pharmacokinetic properties, medicinal chemistry properties, whether it is synthesizable or not, everything you can get these results. Okay. Maybe someone doubt, is it possible when I do all the free web sources to do the analysis is possible to publish? Yes, it's possible. These are the examples. They are using entirely free software tool and get the results and done the publications. Okay, some people. Okay, another one is, sir, I don't have knowledge on the chemistry. I'm interested in the natural product. I want to do the research on the natural product by computer drug designs. What we can do? Simple. We can go this website. It's a natural product atlas website. It's a free tool. Once you design any molecules, or if you found any molecules, you just load your molecules. It will show the similar phytochemicals. This is example. I load the small molecule, give the similar phytochemical report molecule. You can download this molecule and go for phytochemical screening analysis, phytochemical analysis like that. Okay. Some people they want to do the particularly plant-based work. Sir, I'm planning to do the research on the medicinal plant, where I can do the phytochemical of the database. It's free. It's one is a good online tool. It's a Duke phytochemical ethnobotanical database. In this website, you can log in. You can type your what's the plant you want to do the research and search. It will give the entire phytochemical list with ICFT also and where it is available. So easily you can download this my molecule and the CSV file or SMILE format or STF format, everything you can download from this plant. It's a highly authenticated database. You can download the list of the phytochemical report in the plant. So no need to go for article to download the molecules. You can go this website. Easily you can download all the molecules what are available in the plant. Okay. Sir, I found target proteins. I don't know what are the molecules reported in the target protein. I want to know the informations. So you can go this binding TP. It's one website. It's a free online tool. Here we can enter your what are the protein you want to do the analysis. You type the protein name, then run the work. It will ask what are the protein correct name. Once you select the correct protein name, and it will give entire results up to now what are the molecule reported in the target protein and what is the activity, everything it will give. So whether publication or database, everything it will show. So easily you can download all the molecules by STF database. It will give the millions of molecules with the activity. You can download this molecule. It will mainly used for the QSR analysis, pharmacophore analysis, and drug design also. The database will work. So from this information, you can design the potential molecule. What are the molecules are highly active or inactive? Everything it will show. So based on that, you can design the molecules. So this website, how will you go? Okay. This is one of the journal. If you want to really do the docking or in silico studies, you can read this article. This article very clearly explain what are the interaction involved, the docking, hydrogen interaction, what was everything. They explain very simply. If you read the article, easily you can get this idea. From the ideas, we can design the molecules. Okay, this is one of the link. It's a click to drug. In the link, they listed what are the free link is available for drug designing. Like you can designing, docking, pharmacophore, QSR, everything they listed in this free channel. Once you enter this website, you can find a lot of the free online tool, and you can do the research based on these informations. Okay, this is not research. I'm going to explain is actually my institute is ICReDD. They are giving an offer for internship for everyone. So this we can, if you want to do the research in our institute, you can apply this position. They'll give the 1 month to 3 months for internship, accommodation, flight for everything free. You can come and you can learn this new technology in the computer didactic science. Thank you.
Thank you so much, sir. It was incredibly very useful session. Our students have few questions from their end, sir. Can I pose the question, sir?
Yes.
Just a moment. So the first question is, so you have introduced a novel color atom-based drug design strategy that moves behind the conventional method. For students new to CADD, how does this AI and ML approach improve our ability to predict the quality of a hit molecule compared to the traditional structure-based docking?
Who asked the question? Because that is a good question, but that I'm not involved. I'm not include that research in my PPT.
Yeah, we have shared your profile to the students beforehand, sir, so they can come with a homework then.
Okay, so a challenging question. Okay, I will answer these questions. How can I start this? Actually, I'm the computational chemist. Actually, I previously when I was working in the company, mainly we are focusing the structure-based drug design and ligand-based drug design. As of now, the worldwide, everyone they are using only two methods: structure-based, whether they go for structure-based, whether go for ligand-based. But the problem is, once you go for ligand-based drug designs, you can first design the molecules. You don't know that molecule is active or inactive. Maybe there is toxic or non-toxic. Nothing you can do. So first they design the molecule, then they go for docking, and they go for machine learning to predict the activity like QSR, then go for dynamics, then go for ATMET. So this is a long process in computational techniques to find the best molecules. It will take nearly 1 month to find the best molecule before the synthesis. That is one method. Once you go for structure-based method, same thing. Once you design the molecule based on the protein, again go for docking, dynamics, pharmacophore, QSR, then only can best find the best molecules. But I developed this color atom methodology, but I could not share the PPT, but it is very confidential. The reason is I developed the color atom means it's purely based on the machine learning techniques. Machine learning means if I want to design the potential molecule for Alzheimer's disease, for example, first what I'm doing means, as of now, what are the drug reported in the Alzheimer's disease, either active or inactive. Everything I collected from the millions of molecules I collected, then go for machine learnings. I make it the best models. So the model is understand what are the molecules are active, what are the molecules inactive. They have the informations. From the information, I make it the color informations. So color information, once I load my new molecule, it will show the green, it will give the full entire green color. It should be the molecule giving the highly active. Once it giving this red color for the small fragments, it is a non-active. So without information of the docking, pharmaphore, QSR, everything, just design the molecule and load my molecule in this my model. Within second, it will show this fragment have toxic. So immediately I can change the molecules. So I tried this method. Once you entire molecule coming to the green, once I check for in silico studies, it gives a good activity. So I understand this is a new strategy for drug designs. Currently, it is under the process.
Oh, that's a brilliant idea, sir. That is great. Okay. So next question is, you're currently working in Japan and using machine learning to explore the gigantic chemical space. Can the AI actually invent a brand new molecule that never existed before?
Can you ask one second? I could not understand.
Yeah. So can the AI actually, the artificial intelligence can actually invent a brand new molecule that has never existed before? Like will it help us to find a molecule within existing database?
Yeah, actually, that is a very challenging challenging issue there. Actually, I will explain very clear what are the issue on the AI based. See, AI does not about these chemistries. If you are giving the information like if you are giving the 100 molecules, highly active molecules, if you go for machine learning and it give the get the information, and using our some AI code, we can develop the new molecules. It is active, but the problem is that molecules synthesis or not. That is the biggest issue. Yeah, the molecules, as per if the AI is showing the good molecule, once you go for a docking, pharmaco, whatever, it's giving a good activity. I explained. But at the final stage, I could not synthesize the molecules. Even if you go for retrosynthesis, it will give a long scheme. It's very difficult to synthesize. There is no use for that type of method. That's what I'm actually, I don't know, I mentioned the CV or not. That's what I make it the first time, a reaction template.
Yes, we saw that in your
Yes, sir. One minute.
just mentioned.
Madam, some ad is coming. Can you stop this ad? Because
1 minute.
okay, fine. Yeah, that question is, AI surely it will design the potential molecule, but that molecule not possible synthesis, no use. Which means AI does not about the background of the chemistries. So that's what first time in my worldwide, first time I developed the reaction template. Reaction template means I developed the reaction informatics based on our chemistry standard book. We following our chemistry book like Jackley and some of the organic naming reaction book is there. This is a standard book. That is they have the mechanism reactions. So I read the reactions and I know the chemistry, how the product's coming. Based on the reactant A, reactant B, reactant product C, I make it informations. For example, Suzuki reactions. Suzuki reaction is reactant is halides, aryl halides and boronic acids, the CC coupling, and we can go with the products. This is a mechanism of the Suzuki. So I'm making the Python template. It's a reaction informatics. I give the information. What are the aryl groups? What are the boronic acids? Everything written in the smart format. It's a Cheminformatics writing method, smarts. Smiles method is there, smart method is there. So I can write the smarts and write the products, which means actually the product will come there based on the mechanisms. So it won't give the any wrong product, only give the correct product, because I developed the template based on this mechanisms. Once you load this information to the AA, the AA developed the new molecule based on the my template, which means the molecule 100% possible to synthesis. That's what I'm working.
So that's great. You actually teach the AI about chemistry, it seems.
Okay.
Okay, next question is, so since you have a background in pure chemistry, at what point in your career did you realize that learning programming and AI was necessary to stay competitive in emerging technologies?
I think someone known person they asked the question. Actually, to you.
No, sir,
Actually, I'm the chemist. In my PhD, I planning to synthesize the molecules. Actually, we know that chemistry really we have to take. Once you easily we can put the scheme based on this reporter. We can put the scheme. Once your scheme is novel as a chemistry, immediately we can synthesize the molecule, 10 molecule, 20 molecules. Then finally we can take, that's I synthesize 20 molecule, new molecules. Then go for biological activity. 99% all the molecules give you the low score, less activity. If the chemist is the molecule not giving the activity, generally what we'll do, we cannot throw the molecules because we spend a lot of money. So simply we change the data, manipulate, and it is activity. No one can come to cross check. That's what I face. I want to change my career as a computational chemistry and my PhD. So first I learned the computational chemistry for 2 years, and that I go for synthesis, which means before synthesis, I can analyze the molecule by computational techniques. Then I can synthesize the molecule. That molecules, if you synthesize 10 molecules, 9 molecules are active, 1 molecule is non-active. Simply throw it. But without information of computation technique, the 10 molecules are active or nothing. No, many molecules failure. That's the issue. And another issue is, it is actually when I ask our company, synthesis is very toughest to work. Always we have stand in the lab. Always stand in the lab, synthesize the molecules. But computation is nothing. Just write the program and it will give the results and good results. That's what I changed my profile here.
That's a profound thinking.
And I want to share one thing, if you have time or not.
Yes, of course, sir. We have time.
Actually, my institute is ICReDD. They already got 2 Nobel Prizes. Here they installed the robotic synthesis. So we know that a chemistry lab is there. Human will go to there and they synthesize the molecules. But here the robot will do there. The reason is the human always do the error. And a human will only work 8 hours, but robot will work 24 hours. So the robot is installed in the laptop, the robot will synthesize the molecule, but the robot does not know the reactions. What are the chemicals? Nothing. Behind, we are making the reaction template and give the command to the robot where you have to take the reactant A, where you have to take the reactant B, and how many hours it will be react, and where you have to transfer, like everything we are programming. So the robot will be randomly synthesize the molecules and go for column like that. In the column, we know that many molecules will come like that. We don't know which is compound, which is impurity, which is reactant, nothing. Robot does not know. If your peak is coming, giving the product like that. So we are working as a machine learning, and based on the machine learning, we found when the molecule coming from the column, we are predicting the retention time. Once you give the retention time to the robot, that retention time only the molecule will be come out. So it's entirely automatic like that. We developed here like that is the first time.
It's really enthusiastic. So great, sir. Great. So we'll check your website about your, the institution about all this robotics.
Yeah, try to apply. Actually, recently this year they are planning to take more women candidates for the Manabia program. They give you the 1 month to 3 months. So kindly apply and you can come and learn.
Oh, that's great. Definitely be very useful to our students also. Actually, this was my last question, sir. So for the present current project on Hokkaido University, the robotic synthesis. So what is the first step for a student to become a computational chemist? This is from my end. I just want to make my students know about it.
Yeah, actually the issue is in mostly in India. I'm not blaming. There is no subject for learn the computational chemistries. There is no degree. There is no college to teach the computational chemistries. That is the biggest issue. But other countries they already started. So if you want to learn the computational chemistry, you have to read the online tools or YouTube or articles. Very difficult for student means they have to read the some other free online tool. What is the computational chemistry? How we can start everything. It will take 1 or minimum 6 months uh,
To
if
get into it.
get the informations. But I I request to all the chemists to thoroughly read the book, but simultaneously you can learn the computational chemistry because in all the world they are changing the computational to chemistry to computational chemistries.
Yes. Yes.
So you have to learn some advantage.
Yes. Sure, sure. Definitely, we will encourage our students to start learning this kind of advanced techniques also, sir. Okay, so this is all about the question, sir. So we have our HOD ma'am waiting for read the summary of this whole international webinar. 1 minute, sir. I'll hand it over the session to Dr. G.P. Bhavani.
Good morning to everyone. I'm Dr. G.P. Bhavani, Head of the Department of Chemistry. A journey through the emerging technology. Redefining the boundaries of the chemistry. Today, we live with a vibrant map of the future. The day one, the foundation of the modern materials like polymers. We inaugurated our seminar with Dr. N. Muzaffar Khani, who set the stage by exploring the vast world of polymer chemistry. We learned that polymers are the silent engines behind the almost every modern convenience. Dr. Muzaffar Khani sessions reminded us that the chemical engineering of macromolecule is an art of the all materials innovations. The day two, digital revolutions, bio and informatics. You opened by bridging the gap between the test tubes and computer screen. We learned that the modern chemist is no longer restricted to the bench. Through the bioinformatics and informatics, we explored the how data is becoming our most powerful creators. Dr. P. Ravikumar made us to see how algorithms can predict molecular behaviors, allowing us to screen thousands of compounds and seconds, a process that once took years of manual labors. The day three, powering of future, midway through the our focus shifted to the global challenges of the mind of wind. It's actually mind of chemistry. We discussed the critical materials like a wind turbine's efficiency under transformative potentials of green hydrogen. The day four, precisions and standard industrial pharma. Yesterday, Mr. Kumara Raja KKN brought us into the heart of pharmaceutical industries to understand that world quality assurance and regulatory affairs. Precision is the negotiable for perhaps the most important. We exploring how AI and the machine learnings are how now the guardians of product safety and stability. Day 5, AI fraternity, computational chemistry. Finally, today, Dr. Balasubramaniyan Saktive connected all this thread by sharing his research for Hokkaido University, Japan. He showed that AI militates not future technologies. It is currently engine drug discovery. From robotic synthesis to predict modeling, we have seen that next generation chemistry must as a comfortable for the Python as we have with the Pipettes. Thank you. We will hand it over the sessions to Ms. Ashwini for vote of thanks. Thank you.
Good afternoon to everyone joined us online for our international seminar. It's my honor to give the vote of thanks for the fifth day of this week long international seminar organized by the Department of Chemistry at Sri Chandra Prabhu Jain College, Minjur. First of all, we sincerely thank our respected expert speaker, Dr. Balasubramaniyan Shaktivel, Cheminformatics Scientist. Sir, thank you for your sharing valuable knowledge and for giving us a wonderful closure to this week-long seminar. Your session truly inspiring and helped our student and faculty members understand modern developments in our field. We express our heartfelt thanks to our management and Secretary Lalit Kumar Ojay and our principal Dr. N. Sujata for their constant support and encouragement. Our special thanks to our organizing convener and head of the department, Dr. G.P. Bhavani, for organizing this meaningful program and bringing expert knowledge to young minds. We also extend our sincere thanks to Mr. Balaji and the technical team for their dedicated effort in ensuring a seamless and highly quality stream for all our participants today. We also thank all our students and faculty members for attending and actively participating. We hope today's session inspired you, will help you for your future endeavors. Once again, thank you everyone. Have a pleasant afternoon.